Now that a liquid PD-L1 is available, does this impact your treatment decisions and planning, Dr. Hart?
Well, yes, but there's a great potential for doing that. There is right now no other test available other than the tissue test, which we've had for a long time. But looking in liquid for PD-L1 is a really novel approach. So it does open up possibilities for using immunotherapy checkpoint inhibitors in patients where we have not been able to get access to those drugs and not been able to find that the patients could potentially benefit from those drugs. I think that this will allow us to explore a wider range of tumor types and patients that could potentially benefit from checkpoint inhibitors that we have not seen. I mean, there is has been a move in the last few years towards more, and everyone knows, especially with pembrolizumab, it's been opened up as a tumor agnostic treatment for patients that have a high tumor mutational burden. And but not all patients have that. That's not always available. And looking for high levels of PD-L1 in a liquid biopsy is a very novel approach, and I find that very interesting and may potentially enable us to expand the use of these drugs and have other patients potentially get very significant benefit from the use of them.
Thank you. Dr. Reeves?
I think that, from my perspective, I think this helps us address a number of research-related questions.
Mm-hmm.
While PD-L1 is a marker, and at this point, I guess perhaps the best we have at helping to predict immunotherapy effectiveness, not all patients with high PD-L1 respond well to immunotherapy, and patients with zero PD-L1 can also do well. It's just their chances aren't quite as good. So being able to have the PD-L1 up front, I think, raises a number of possibilities for future research questions. Basically just pushing us on a pathway towards more immunotherapy or away immunotherapy, looking at other approaches, particularly in the first line. I think some trials are just starting to address that or use other ways of manipulating the immune system besides the PD-1, PD-L1 axis, and trying to determine whether that will make a difference. I think the thing that we need to get away from is just giving everybody immunotherapy, because when you do that, you're altering that immune environment for whatever the next treatment happens to be. If that's in the patient's best interest, yes, you want to do it. But I think we're gradually uncovering situations where either because of co-mutations or other influencers on the immune environment, that upfront immunotherapy may not be the best solution for each patient.
Wow. Thank you for sharing. Dr. Walker?
I again agree, Dr. Hart and Dr. Reeves. This is such an exciting time with the immune era of treating cancers. And one thing that's well known about PD-L1 expression is that it can upregulate and it can downregulate. Certain treatments can upregulate it and downregulate it. It's not just going to remain the same. And you don't want to not give somebody immune therapy because that could benefit from it, because you're talking about the durability of living. But by the same token, if somebody's not going to benefit from it, you don't want to subject them to the potential significant immune toxicities. So it just opens up as sort of the first step and a foundation to also build upon other immune biomarkers, as Dr. Reeves indicated, to then better select patients. And just another quick clinical comment. For example, it's been well known in breast cancer, you can have ER positive, you can have HER2 positive, or you can have triple negative. And a third of the time, once a cancer progresses, it switches to something else. So in breast cancer, you could be ER positive in the primary breast tissue. You could be HER2 positive in the brain. You could be triple negative in the liver. And if you just rely on one biopsy site, you may miss PD-L1 in breast cancer, which does have a survival benefit.